Good evening, everyone. My name is Marasha Jakit. It is November 22nd, 2025, and I will be doing my presentation on the DNA organization, replication, and repair. So the learning objectives for this presentation will be to describe the hierarchical organization of DNA from the nucleosome to the mitotic chromosome, explain the semi-conservative mechanisms of DNA replication and the specific roles of key enzymes like helicase and polymerase, compare and contrast the major DNA repair pathways such as mismatch repair, base excision repair, and nucleotide excision repair. The last objective will be to connect defects in these fundamental processes to the pathology of human diseases, including cancer and aging. So let's first talk about the genetic blueprint. The human genome contains over 3 billion base pairs. DNA from a single cell, if uncoiled, is over 2 meters long. And when it is organized within a nucleus, it is only 5 to 10 in diameter. So in this presentation, we will explore the 3 core processes that manage this blueprint. Organization, so how is it packaged? Replication, how is it copied? And repair, how is it protected from damage? So part 1 is all about DNA organization. What is chromatin? Chromatin is the dynamic complex of DNA and specialized proteins. The primary proteins are histones, so H1, H2A, H2B, H3, and H4. Histones are highly basic proteins which are rich in positively charged lysine and arginine. These positive charges allow them to bind tightly to the negatively charged phosphate backbone of DNA. This organization is not static. It must be constantly remodeled to allow for gene expression and replication. And there's images to the right or to the left of the chromatins and the histones. So the nucleosomes, they're often referred to as beads on a string. They are fundamental repeating unit of chromatones. The bead is the nucleosome core, a protein octamer that has 8 histones, 2 copies of each, H2A, H2B, H3, and H4. The wrap has 147 base pairs of DNA, which is 1.75 times around the histone core. The string is a short segment of linker DNA that connects one nucleosome to the next. And the clamp histone H1 binds to the linker DNA and the core, which stabilizes the structure and facilitates the next level of compaction. So we are now going to talk about the higher order coiling to form the chromosome. So from solenoid to chromosome, the beads on the string is coiled into a more compact structure. So the solenoid are nucleosomes that are wound into a tight helical coil. Histones H1 are the most essential for this. Loop domains, the 30 nm fiber is then formed into loops which are anchored to a central protein scaffold. In the mitotic chromosome is the loop structure that is further compacted and coiled to form the highly condensed structure seen during cell division, as you can see in the image to the right. So the multi-level packaging process achieves a compaction ratio of up to 10,000 folds. So chromatin is not static. It has 2 distinct functional states that are crucial for gene regulation. So euchromatin is basically known as open for business. It is less condensed. It is an open structure. It is active. The DNA is accessible to RNA, polymerase, and transcription factors. The heterochromatin is closed for business, so it's very highly condensed. It is a closed structure, 30 nm fiber and beyond. It is inactive, meaning it is silent or quiet, and the DNA is inaccessible to the transcriptional machinery. The cell converts regions between these states to control which genes are turned on or off. So you can see the images to the right. The heterochromatin is a lot more of a closed structure, and the euchromatin is a more open structure. So part 2 will be about DNA replication. Replication is a process of copying the DNA blueprint. This process occurs during the S phase of the cell cycle. And this mechanism is known as semi-conservative. The double helix unwinds, and each parental strand serves as a template for a new daughter strand. This results in 2 identical DNA duplexes that contain an old strand and a new strand. This ensures the faithful transmission of genetic information from one cell generation to the next. And the replication fork has some key players. The helicase unwinds the DNA double helix. The single-strand binding proteins, also known as SSBPs, prevent the separated strands from reconjoining. Primase synthesizes a short RNA primer to provide a 3-OH starting point. DNA polymerase is the master builder that synthesizes the new DNA strand, and it has a proofreading function to ensure accuracy. And DNA ligase glues the DNA fragments together. You can see the image to the right. So DNA polymerase is the enzyme that synthesizes the new DNA strands, and it has 2 fundamental rules. The first rule is that it needs a primer. It cannot start a new chain from scratch. It can only add new DNTPs or deoxyribonucleotides to the 3-OH groups of a pre-existing strand, which is the RNA primer. Rule 2 is that it has to have a one-way street. It only synthesizes new DNA in a 5-3 direction. It reads the template strand and builds the new strand. So it reads the template strand from 3-5 and builds the new strand from 5-3. These 2 rules create a significant challenge because the 2 strands of the DNA helix are anti-parallel. So to solve this problem, this creates a problem because the 2 strands are anti-parallel. So the leading strand synthesized continuously. The polymerase follows the unwinding helix, and the lagging strand is synthesized discontinuously in the direction away from the replication fork. So the leading strand and the lagging strand is how they help with the directionality problem. The lagging strand synthesizes in short bursts called the Okazaki fragments. Each of these fragments require its own RNA primer, and the DNA ligase joins the fragments together into a single continuous strand. The end replication problem: on the lagging strand of a linear chromosome, the final RNA primer at the very end cannot be replaced with DNA, so there is no 3-OH to build upon. This means that the chromosome gets shorter with every cell division. So the solution are telomeres, which are long, repetitive, non-coding DNA sequences that act as a disposable cap on the end of the chromosomes. The telomerase is a special enzyme that carries its own RNA template. It extends the telomeres, counteracting the shortening. So it is highly active in stem cells and 90% of cancer cells, which gives them limitless replicative potential. Our DNA is constantly under attack. The genome is constantly damaged. Failure to repair this damage leads to mutations, which are the root of cancer and other diseases. Sources of DNA damage includes internal, which is also known as endogenous, which include replication errors, reactive oxygen species from metabolism, and spontaneous deamination. Another source of DNA damage is external, which is exogenous, which includes UV radiation such as sunlight, ionizing radiation such as X-ray, and chemical mutagens such as tobacco smoke. The DNA repair toolkit has some key pathways. So mismatch repair, also known as MMR, is the function for that is a spell checker that fixes errors made during DNA replication. The mechanism of MMR is to identify the mismatches, determine which strand is the new one or the incorrect one cuts out the patch of that new strand, and the DNA polymerase fills in the gap. Another repair toolkit is the base excision repair, which functions as fixes single damaged bases like those from deamination or oxidation. It acts like a scalpel, so the DNA glycosylase cuts out only the damaged base. Other enzymes then remove the rest of the nucleotide, and polymerase or ligase patch the tiny hole. And the nucleotide excision repair fixes bulky lesions that distort the DNA helix, most famously the thymine dimers caused by UV light. It acts like a patch, so the nucleotide excision repair complex cuts the DNA strands on both sides of the damage, removing about a 30 nucleotide patch. Polymerase and ligase then fill and seal the gap. So when repair fails, certain things can happen. Examples include xeroderma pigmentosum, which is a defective pathway from the nucleotide excision repair. Symptoms include patients that cannot repair the DNA damage caused by sunlight. Results include extreme sun sensitivity with blistering and a greater than the 1,000-fold increase risk of developing skin cancer. Another problem can include Lynch syndrome, which is a defective pathway from the mismatch repair. The symptoms include the effects is replication errors are not corrected, leading to a rapid accumulation of mutations, which results in a very high hereditary predisposition for colorectal, uterine, and other cancers. Example here in the image is an example of xeroderma pigmentosum. So our 5 multiple choice questions. The fundamental repeating unit of a chromatin consisting of DNA wrapped around a histone octamer is called centromere, solenoid, nucleosome, or telomere. Number 2, during DNA replication, the lagging strand is synthesized as a series of short fragments known as Okazaki fragments, promoter sequences, start codons, or introns. Which enzyme is responsible for unwinding the DNA double helix at the replication fork? DNA ligase, DNA polymerase, primase, or helicase. A patient who is extremely sensitive to sunlight and has a high incidence of skin cancers found to have a defect in their DNA repair system. Which pathway is most likely affected? Mismatch repair, nucleotide excision repair, base excision repair, or homologous recombination. The enzyme responsible for maintaining the length of chromosome ends, which is often reactivated in cancer cells, is called topoisomerase, telomerase, connectochore, or nuclease. Here are my references. Thank you so much for watching my presentation. I hope you guys have a great day.